Hey, I'm Blake Sproles, and this week I've been working with Professor Gazdik on our project, which is studying Mycobacterium tuberculosis and CAMP. So far it's been really enjoyable. I've learned all sorts of stuff. It's kind of given me some experience working here in the lab, figure out if that's really what I want to do as far as a career in science or not. I really enjoyed it, so it's something I may look into. Kind of like the little medical aspect of it as well, working with the bacteria and tuberculosis aspect of it. So it's been great, and I would really like to thank Dr. Gazdik for working with me and for kind of giving me some insight on everything. And it's been a great program, and I'm just very thankful for being accepted into it.
And it's actually been really great having him in the lab. It's nice to get an exciting group of students in here that actually are having a good time and wanting to learn and doing a lot of work in the lab. And we're hopefully getting some data today, so we'll see how the project goes by the end of the week.
So my project dealt with studying the Mycobacterium tuberculosis bacteria. Mycobacterium tuberculosis is the bacteria that causes the tuberculosis infection. What we're studying is the cyclic adenosine monophosphate concentrations that each bacteria secretes. We're using three bacteria for ours, E. coli, Smegmonas, and Bovis BCG. E. coli is kind of like the basic bacteria, so it's not anything in like relation to Mycobacterium tuberculosis. Smegmonas is a slower growing bacteria. It does have some of the vaccines, or not the vaccines, but some of the similar DNA as the Mycobacterium tuberculosis, so that's why we're using that one. And the Bovis BCG is actually the vaccine strain that they use in some of the treatments for Mycobacterium tuberculosis. So these are the bacteria we're using, and what we did was we kind of put them through various tests to get cyclic ANP values, which is the cyclic adenosine monophosphate I told you about. So what we did was we grew different cultures of each bacteria. From these we took spectrophotometer readings, and we got our growth curves for the bacteria. After we did that, we also took various samples from each time period, and from those we got secreted and intracellular CAMP values. Here you can see our secreted values. The Smegmonas, which is the red, has a much more defined growth and decline in their values. E. coli is a little bit more just kind of varying, nothing in particular. And again, in the intracellular, you can see the Smeg has a much more defined growth and secretion, growth and declining in the intracellular CAMP. Here we've actually got the CAMP concentrations. You can see here that the Smeg is about 4.5 times more than the E. coli and the CAMP concentrations. So the BCG that we're studying, we actually have not got any values for it yet because it's such a slow growing bacteria. That one you have to kind of analyze in days rather than hours like we can do with the other two bacteria. So what we're hoping to do is once we get our data for the BCG, we'll take it and we'll compare it with the Smegmonas and the E. coli values as well. We're expecting there to be a little bit of correlation between the Smegmonas and the BCG, but we don't want to know until we get our data for that for sure. But once we do get that, what we're going to do is we're going to put all the bacteria through various environmental changes, such as decreased pH, starvation, low CO2 values, and the presence of fatty acids. The reason we're doing that is to test the bacteria whenever they're inside of the body, because those are the different factors they go through whenever the Mycobacterium tuberculosis enters inside the body rather than outside. So that's what we're looking to do in the future. So it's going to be something else. I've got a pretty heavy class schedule in the fall, so what I'm going to do is kind of see if I can find a time balance and stuff like that. Once I get that, she told me I can always pop my head in every now and then check on the project. And she said once we get to the spring, I'm going to be working with another student, and we're going to work with the future work by testing the different environmental changes. If we do get some good values and data for the different environmental changes and the BCG and stuff, we may go ahead and try to get the work published. Yeah, it's been a great 2 weeks though. Really enjoyed it.